Hello everyone, this is a presentation from Group 1, Commerce, on protein structure characterization with mass spectrometry. Today we are going to have a look at introduction, recognizing conformational changes between different protein states, protein mass mapping of a protein or protein complex, time-resolved protein mass mapping of virus, followed by chemical cross-linking and chemical modification, and finally conclusion. Coming to the introduction, mass spectrometry, which is abbreviated as MS, is now commonly being used to determine both the primary and higher order structures of proteins. Protein mass mapping combines enzymatic digestion, mass spectrometry, and computer-facilitated data analysis in order to produce and examine proteolytic fragments. This is done for the purpose of identifying proteins and more recently for obtaining information regarding protein structure. Let's have a look at the protein identification. Protein identification process involves a series of steps in which the incubation of sequence-specific proteases is done with the protein of interest and mass analysis is performed on the resulting peptides. And the produced fragmentation pattern is then compared with the patterns predicted for all proteins, which is followed by the statistical evaluation of matches. Similar to this, the higher order structure of a protein can also be evaluated when mass mapping techniques are combined with limited proteolytic digestion. And the sequence specificity of the proteolytic enzyme plays a major role in the application of mass spectrometry to protein structure. And this sequence specific protease enzyme can be capable of reducing the number of fragments that are produced, and concomitantly it improves the probability for statistically significant matches between the observed and predicted fragment masses, and it is also capable of reducing the opportunities for spurious matches. Another factor in this is the accessibility or flexibility of the site to the protease, which plays a major role in the analysis of protein structure. In this case, ideally, only a subset of all possible cleavages are observed owing to the inaccessibility and or inflexibility of some sites due to the higher order structure of protein. This is an example given which is illustrating the use of limited proteolytic cleavage as a probe of protein structure. Here arrows indicate flexible sites that are accessible to proteases which are exposed on the surface of a hypothetical protein. If a sequence-specific proteases were used, the marked sites would also have to contain the protease recognition sequence in order to sustain the cleavage. Mass analysis of all proteolytic fragments together yields the cleavage map from which we can obtain an information on the protein structure. Proteases that can cleave at hydrophilic sites are preferred in structural analysis of proteins because on the surface of proteins there is a greater abundance of amino acids with hydrophilic side chains. Trypsin, which cleaves at basic sites, and V8 protease, which cleaves at acidic sites, are considered to be good choices for this. And all the reaction conditions must be controlled in order to produce only limited proteolysis so that the cleavage pattern reliably reports on native tertiary structure. The cleavage of a single peptide bond, which can destabilize the protein, causing local structural changes or even global unfolding, whereas the subsequent protease cleavage reactions would not inform on native structure. Protein mass mapping can also be used to probe the quaternary structure of multi-component assemblies such as protein-protein complexes, protein-DNA complexes, and even intact viruses. Chait and co-workers were the first to apply the limited proteolysis and MALDI, which is called matrix-assisted laser desorption ionization, mass spectrometry on the study of a multi-component biomolecular assembly. In their studies, this combined approach was used to analyze the structure of the protein transcription factor, called MAX, both free in solution and when bound to an oligonucleotide containing its specific DNA binding site. The common feature that was observed when analyzing either protein-protein complexes or protein-DNA complexes is that the protease provides a contrast between the associated and unassociated states of the system. The formation of an interface between a protein and another macromolecule will protect or otherwise accessible sites from protease cleavage and therefore provides information about residues that form the interface. Coming to the process of recognizing conformational changes. In order to recognize simple conformational differences that are present between different protein states, protein mass mapping can be used. For example, X-ray crystallography data has shown that the protein calmodulin undergoes conformational changes in the presence of calcium. It has been proposed that the calcium binding activates calmodulin by exposing hydrophobic residues which are present near the two ends of the central helix. The resulting mass maps that are obtained from digest by trypsin, chymotrypsin, and pepsin all have demonstrated that the protein had undergone a tertiary structural change in the presence of calcium. This figure shows the MALDI mass spectra that is obtained from the trypsin digestion of calmodulin both in the presence and absence of calcium. By comparing the two mass spectra, differences were observed corresponding to cleavages within the central helical region of calmodulin represented by black dots, which are not observed in the presence of calcium represented by yellow dots, indicating that there is a tertiary conformational change of calmodulin that is taking place in the presence of calcium. Both the spectra and the ribbon drawing that are illustrated in the figure were shown with the cleavage sites that are present in the calcium calmodulin complex with black dots and those that disappear upon addition of calcium with yellow dots. Electrospray ionization mass spectrometry, which is abbreviated as ESIMS, has also been used to monitor protein folding and protein complexes. Electrospray mass analysis can be used to distinguish between native and denatured conformers of a protein. For example, two charged state distributions were shown in this figure, which depicts a protein's less charged native form and the more highly charged denatured conformer. The difference between the spectra is due to the additional protonation sites that are available in the denatured form. This phenomenon is demonstrated for the protein called fibronectin, where the charge distribution is maximized at the lower charge states, that is greater than or equal to 7+, and the denatured protein has a distribution maximized at a higher charge state, that is 10+. This ESIMS technique is a simple but highly sensitive and informative method which is used to characterize the functional shapes of proteins, that is either globular or extended, prior to more material intensive and time consuming spectroscopic or crystallographic studies. This figure illustrates the theoretical mass spectra of two different populations of proteins, for example native and denatured forms. The middle peak represents a theoretical protein folding intermediate that is partially folded, and the use of hydrogen or deuterium exchange in order to study the conformational changes in proteins or protein-protein interactions has been primarily performed using ESIMS. The concept of this approach is relatively simple, that the amide protons within the portion of the proteins in close inter or intramolecular contact may form hydrogen bonds and will have different exchange rates relative to other more accessible regions of the complex. By monitoring this amide hydrogen exchange, information can be obtained on the non-covalent structure of a protein by itself or in a protein complex. In order to monitor the protein folding reactions, the combined application of ESIMS with NMR spectroscopy using HD exchange is more powerful. Let's have a look at protein mass mapping of a protein or protein complex. The cell cycle regulatory proteins called CDK2 and P21B were examined using protein mass mapping, which exploits the high mass accuracy, resolution, and sensitivity of MALDI-MS. Initially, proteolysis reactions were performed for one component before and after formation of a multi-protein assembly, and then proteolysis reactions were performed for the complex, and the reaction products for both experiments were analyzed using MALDI-MS. This figure depicts the probing of protein or protein interactions using proteolysis and MALDI-MS. There is a schematic view of the key concepts on the left side of the figure. On the top of it, a protein with two cleavage sites was shown, yielding 5 fragments after limited digestion, whereas at the bottom, in the complex with protein X, one of the site is protected, yielding fewer fragments. However, protein X also produces some fragments which are designated as XF, and the actual results obtained were represented as a histogram and were shown on the right side of the figure, indicating the region of interaction of P21B with CDK2 in a 24 amino acid segment, which was later confirmed through crystallographic data on a homologous system. In studies of multi-subunit protein complexes, the resolution of multi-MS may not be sufficient in order to resolve all proteolytic fragments. In these cases, selective isotope labeling can be employed in order to identify the fragments of individual subunits. In addition to that, high-throughput mass spectrometric protein complex identification, which was abbreviated as HMSPCI, has been recently used to identify protein-protein interactions in yeast. In this process, carefully selected bait proteins were epitope tagged and attached to a column. An affinity purification process was carried out using whole cell lysates. One-dimensional SDS page analysis was then used to separate the resulting immunoprecipitants, and the stained bands are excised and digested, followed by mass analysis. Due to the likelihood of multiple proteins being present in a single stained band, LC-MS was used to analyze the proteolytic peptide and also allows for high throughput analysis. Following this analysis, a network map can be created in order to illustrate various proteins. This figure illustrates several hypothetical interactions that could exist between a network of proteins. For example, one protein may interact with several downstream partners, resulting in a wide array of effects. These experiments are also clearly valuable for illustrating the dual functionality of proteins, such as many proteins involved in the DNA repair process have also been shown to have roles in DNA replication. In addition to this, it appears that HMSPCI can be used to identify protein complexes from a range of subcellular components including cytoplasm, nucleus, plasma membrane, and mitochondria. These studies are being conducted as part of a systemic approach to proteomics that will help define protein networks and pathways involved in cellular functions. Coming to time-resolved protein mass mapping of viruses. Viral analysis is a logical step in the development of protein mass mapping techniques, since viral caspids represent the non-covalent quaternary association of protein subunits. Since proteolysis is performed in solution, different conformers can be detected, hence this method can contribute to an understanding of the dynamic domains within the virus structure. Limited proteolysis or MALD-MS experiments were performed on human rhinovirus 14, which was abbreviated as HRV14, and flockhouse virus, that is abbreviated as FHV. HRV14 is a causative agent of the common cold, and it is a member of a family of animal viruses called the picornaviruses. This HRV14 virion consists of an icosahedral protein shell or viral caspid surrounding an RNA core. The caspid is composed of 60 copies, each of 4 structural proteins, that is VP1 to VP4. Based on crystal structure data, VP1, VP2 and VP3 compose the viral surface, while VP4 lies interior at the caspid or RNA interface. Like HRV14, FHV is also a non-enveloped icosahedral RNA animal virus. The mature protein coat or caspid is composed of 180 copies of beta protein and gamma peptide, which were shown in this figure. In protein mass mapping experiments, viruses undergo limited proteolysis followed by mass analysis of the proteolytic fragments. In time-resolved proteolysis, the study of protein caspid mobility was done. It was expected that the reactivities of viral particles to different proteases would reveal the surface accessible regions of the viral caspid. Observation of digestion fragments resulting from internal protein regions was initially perplexing, but after further examination of these results along with X-ray crystallography data, it was determined that portions of the internalized proteins such as VP4 are transiently exposed on the surface of the virus. This figure shows the results obtained from the trypsin dissection of the time-resolved proteolysis of human rhinovirus. Interestingly, it was found that the fragments from VP4 were first observed in the limited digestion of the virus. These experiments clearly demonstrate the dynamic nature of the viral caspid. Chemical cross-linking and chemical modification. Both these approaches arise from the fact that MALDI has proven to be a powerful method with which we can characterize covalent post-translational modifications. These chemical cross-linking studies involves the treatment of proteins with cross-linkers prior to digestion, whereupon adjacent protein subunits are covalently attached to one another, and the resulting proteolytic fragments are considered to be good indications of the overall tertiary and or quaternary protein structure. More common than structural studies, chemical cross-linking has been employed to determine the stoichiometry of oligomers with MALDI-MS analysis. This figure is an example to show how cross-linking of a non-covalent protein complex, which was on the top, can alter the MALDI mass spectrum by providing the number of subunits in the complex, such as monomers, non-covalent dimers, and so on. Whereas the cross-linked protein complex, which was on the bottom, produces a MALDI mass spectrum containing a cross-linked trimer. MALDI-MS analysis of a complex helps in determining the molecular mass of individual proteins. In this process, a cross-linking agent such as glutaraldehyde is added that reacts with epsilon amino group of lysine and form cross-linking chains. And finally, the reaction is halted by the addition of a MALDI matrix. A simple illustration of such reactivity for a viral protein after being exposed to acetylation, resulting in limited chemical reactivity, was shown in this figure. And the produced MALDI mass spectrum of a chemically modified viral protein from an intact virus can be used to determine the sites of reactivity, which further provide information about the surface exposed topology of the virus. Coming to the conclusion, this paper illustrates the utility of combining proteolysis as well as other chemical modification methods with mass spectrometry analysis that are used in structural studies of proteins. The MS-based probing methods are particularly well suited to provide rapid access to low-resolution maps that are used to guide subsequent high-resolution studies. And the high-throughput proteomics techniques such as multi-dimensional protein identification technology are being developed, and that will provide a global view of an organism's proteome. And these studies can eventually be used with interaction mapping methods in order to elucidate the individual residues that are involved in these protein complexes. And finally, I would like to thank my group members for their support and Dr. Maya Nair for giving me this opportunity.